Neurons in the brain, or ganglia, are responsible for transmitting sensory information. Sensors such as eyes detect external and internal stimuli, light, heat, blood pressure, etc., and transmit the signals they receive to the brain and ganglia where the signals are processed. Most of the neurons' organelles are in the cell body. Signals are received from branched extensions called dendrites and transmitted through axons. The tip of an axon is a junction called a synapse. Here signals are transmitted from neurons to neurons. Chemical messengers called neurotransmitters pass information from the transmitting neuron to the receiving cell. Sensory signals are sent through neurons by the exchange of ions through the plasma membrane of the neuron. The exchange of ions creates a membrane potential. A neuron that is not sending a signal is at its membrane potential, which is usually between -60 and -80 millivolts. This membrane potential is maintained by the exchange of sodium and potassium ions, which are pumped across the membrane by sodium-potassium pumps. In addition to sodium-potassium pumps, other protein channels such as sodium pumps, potassium pumps, and gated ion channels that help maintain the flow of ions. There is always a higher concentration of sodium outside the cell and a higher concentration of potassium inside the cell. The reason that the resting potential is not at -50 millivolts is because there are more potassium pumps than sodium pumps. Ion-gated channels open and close in response to a stimuli. Opening gated potassium channels allows potassium ions to diffuse out of the membrane. This causes hyperpolarization, which makes the charge inside the neuron more negative and increases the magnitude of the membrane potential. Hyperpolarization can result from any outflow of positive ions and inflow of negative ions. On the contrary, opening some other ion channels, such as sodium ion channels, can cause depolarization, during which the inside of the membrane is less negative and the magnitude of the membrane potential is less. Gradient potential results from a shift in the membrane potential and has a magnitude that varies with the strength of the stimulus. Gradient potential induces a small electrical current that leaks out of the neuron as it flows along the membrane. Therefore, this type of potential decays with distance. If a depolarization shifts the membrane potential significantly, the result is a massive change in membrane voltage called an action potential. Action potentials have a constant magnitude and can spread along axons, making them well suited for long-distance signaling. Action potential is caused by voltage-gated ion channels, which open or close when the membrane potential passes a particular level. In other words, a threshold. If a depolarization opens voltage-gated sodium channels, the resulting flow of sodium ions into the neuron will cause further depolarization, which will then cause more voltage-gated sodium channels to open. The result is a process of positive feedback that triggers the rapid opening of all voltage-gated sodium channels. There are 5 stages to how a voltage-gated ion channel can generate an action potential. In the first stage, the resting state, only potassium channels are open, and most voltage-gated sodium and potassium channels are closed. During depolarization, a stimulus depolarizes the membrane and some gated sodium channels, open, allow sodium atoms to diffuse into the cell. The inflow of sodium ions causes further depolarization and a positive feedback. Once the threshold, which is -55 millivolts, is crossed, the positive feedback cycle causes the inside of the axon membrane to become more positive with respect to the outside. This is the rising phase. In the following phase, voltage-gated sodium channels inactivate and most voltage-gated potassium channels open. The opening of voltage-gated potassium channels allow potassium ions to flow out of the neuron, making the inside of the neuron negative again. In the last phase, the undershoot, the sodium channels close, but some potassium channels remain open. As the potassium channels close and the sodium channels become unblocked, the membrane returns to resting state, and the cycle starts over again. The depolarization-repolarization process is repeated across the axon as the rising phase in one part of the axon causes depolarization in neighboring parts. There is a refractory period during which the sodium channels are not able to generate another action potential for a period of time. This sets the maximum frequency at which action potentials can be generated. Signals are transmitted from neurons to neurons through synapses. An action potential arrives at a synapse and depolarizes the presynaptic membrane. The depolarization opens voltage-gated calcium channels. The elevated calcium concentration causes synaptic vesicles to fuse with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. Finally, the neurotransmitters bind to the ligand-gated ion channels in the postsynaptic membrane. This triggers the opening of other channels that allows the diffusion of sodium and potassium ions. At some synapses, the ligand-gated ion channel is selectively permeable to both sodium and potassium. The depolarization by this brings the membrane potential toward a threshold and is called the excitatory postsynaptic potential, EPSP.